Hello students, welcome to Learner's Planet. Let us continue with the topic tissues. And now just for a brief introduction. In the previous session what we have done is the introduction of the tissues and we have compared the tissues of plants and animals. In the last class we had started with the plant tissues and we ended up with the meristematic tissues. Let us continue the topic, that is plant tissues itself. Meristematic we have done. Now let us begin with the permanent tissues. So before we go in depth of this particular topic, let us start by briefing the idea about the same. So basically permanent tissues, they are derived from meristematic tissues only. You know what? You remember what we said about the meristematic tissues? The meristematic tissues are those which are having the highest cell division capacity, and due to this cell division capacity, these cells, they tend to divide continuously and thus they support the growth of the plants. So on the basis of location where these meristematic tissues are present, they are of three kinds: apical, lateral and intercalary. So all these are supporting the growth of the tissues. Now, don't you think that if these tissues are continuously dividing and they are forming the new organs, or they are just increasing the height of the plant, then how the other functions are being performed? As we know that there are vegetables, there are different kind of food material, flowers, everything are growing inside, and all these need support, all these need protection. They need different kind of processes to occur for the maintenance of growth also. So if these meristematic tissues, they keeps on increasing their supporting the growth of the plants, then how these other functions are performed? So actually what happens is, meristematic tissues, they are dividing and supporting the growth. Besides this process, some of the meristem, it keeps on differentiating. It undergoes some transformation processes, and thus after this they are actually attaining a particular shape, a particular function. They become specialized for a particular function. So such type of process taking place in these meristematic tissues is known as differentiation. So after this differentiation, what will happen? They will form the permanent tissue. So this is how permanent tissues are actually formed. They are derived from meristematic, but how? It is by the differentiation process. Now differentiation process, it is in terms of what? It is in terms of shape of cells, structure of the tissue, regarding its cell wall properties, regarding its you can say like a special function, and so on. So once these properties are attained by these particular meristematic tissue, they convert into permanent tissue. And after converting in this form, these type of cells or the tissues, they lose their power of cell division. So therefore we always say that permanent tissues, they are those which are non-growing tissues of the plant. So these non-growing tissues, actually they have lost the power of division, cell division, but they have attained definite structure and a special function so as to support the mechanical structure and strength of the plant. So now can you think, once these permanent tissues are derived from meristematic tissues, then what could be the base of its differences? They are going to be differentiated from each other. There are many differences. Let us say, let us quote it out here. It is in terms of shape. Meristematic tissues, they are having varying shapes, but permanent, they are having a specific definite shape now. Then differentiation, meristematic tissues, they do not differentiate, but if they differentiate, they will take the shape of permanent tissues. Then comes vacuole. The vacuole is either absent in meristematic or it is so small and few in number. But in the case of permanent tissues, it is a large vacuole is present. Now you remember what is the function of vacuole? It is the storage of various food material or the other components. Now if the vacuole it has taken the maximum part of the cell, then what will happen to the nucleus? It will become inconspicuous. It is pushed towards the side of the wall. Then further, intercellular space. It varies in case of permanent tissues, whereas it is absent in the case of meristematic tissue. Cell wall made up of cellulose in permanent as well as meristematic tissues, but in meristematic tissue it is thin, whereas in permanent the thickness will vary according to the type of the tissue. Then comes the growth point. Of course, meristematic are the growing tissues, but permanent, they do not grow. They have lost their cell division capacity. But these cells are showing the metabolism, and these metabolic functions, they are supporting the special functions of the permanent tissue. In the case of meristematic tissue, they are highly active in terms of metabolism. But permanent tissues, the rate of metabolism is slow. But of course, it supports the formation of different type of things like crystals. Now these crystals are absent in meristematic tissues, but of course they are present in the permanent tissues. Not only crystals, the other structures are also produced, which are known as secondary metabolites. So that is the function of permanent tissues. Then meristematic tissues are 100% the living cells, but permanent, they may be living, they may be dead, they may be a combination of both or vice versa. Then in the case of meristematic tissue, these are supporting the growth, but the function of permanent tissue is what? It is providing protection, support, photosynthesis, then conduction of various material, etc. Now, as we are seeing that these permanent tissues, they are having different type of functions. So definitely, if the function is varied, then of course the structure and its various structure should also vary. So on the basis of such things, these are of different kinds again. Permanent tissues, they can be simple or complex. Now let us just have a brief outlook of the different types of plant tissues again. Plant tissues, they are majorly of two types, meristematic and permanent. Meristematic are those in which the cells have highest cell division capacity. So they can be apical, lateral or intercalary. And all this part we have discussed in our previous session. So now let us come to the permanent tissue. Permanent tissue, they are mature cells, but they have lost the power of cell division. Now, they are of two kinds, depending upon the type of cells present or which are constituting the type of tissue. So, if the tissue, permanent tissue, is composed of a single type of cell so as to perform a particular function. Which type of function? Protective and supporting kind of function. So then, if they are composed of single type of cell, they are known as simple permanent tissues. Further, these are of different kinds. Now we have quoted here function like protective function, supporting function, then storage type of function. So on this basis that what type of function is being supported by these simple tissues, they are of three kinds, parenchyma, chlorenchyma and sclerenchyma. So these tissues vary from each other in terms of its structure, its basic features and its function. So all this we will study further. Besides this, the complex tissue are also known as conducting tissues or the vascular tissues. Now these are composed of more than one kind of cell, but they support the same function. Which function? Conduction. Conduction of various kind of material. So further, what type of material is required to be transported? It could be water, minerals or food. So water and mineral is being transported from soil to the different parts of the plant. So that is, the direction should be from soil to the upper parts, and that function is performed by the xylem tissue. Besides, if food is to be transported, then where it is located? It is synthesized by the leaves. So from leaves, the food is to be transported to different sites, like its storage site or its utilization site, where it is required. So such transport is actually supported by the phloem tissue. Now let us study one by one each kind of permanent tissue. Let us begin with the simple permanent tissues. So these simple permanent tissues, as we have already quoted, that these are those particular tissues which are composed of cells which are structurally and functionally similar. So these cells, they are the group of similar type of tissues which are assigned a particular special function. Now what type of structural cells they are and what type of function they are supporting, on that basis they are of three types: parenchyma, chlorenchyma and sclerenchyma. So let us start with the parenchyma tissue. We said that these parenchyma or other type of simple tissues, they vary from each other in terms of its structure and its function. So let us study about these particular differences, or let us study the basic features of parenchyma tissue, and then later we will compare it with the different kind of simple tissues also. So here the first point to be compared is shape. These cells, they are polygonal. That means they are having multiple end sides, or they can be oval or spherical, but when they get mature, they become polygonal. So these type of tissues, as we are saying, they are having either polygonal shape, or they are oval, or they are spherical. Do you remember what was the shape of meristematic tissue? The meristematic tissues are having various type of shape. Like they can be oval, they can be spherical, they can be rectangular, they can be cylindrical or polygonal, etc. So the shape is changing in case of meristematic tissue. Here, when we study about parenchyma, a simple tissue, then the shape is generally definite. Either it is oval or spherical or polygonal, depending on the function they are being performing there, and of course the age of the cell. Then comes the cell wall. In the case of parenchyma tissue, cell wall is thin and it is made up of cellulose. Now here, as we say that they are having thin cellulose, then if we compare it with the meristematic tissue, there also we said that meristematic tissues are having thin cellulose cell wall. So if both are having thin cellulose, then what is the difference? Comparatively, parenchyma is having more deposition of cellulose as compared to the meristematic, but it could not be considered as thick, as it is of course thin, but thicker than that of the meristematic tissue. So you can see here in the diagram also, these are having this is the cell wall which is shown here. Here cell wall is shown here, and it is the boundary line which is present and is thicker than that of the meristematic tissue. Now these cell walls, these parenchyma tissue, they are having pits in between. They are adjoined with the surrounding tissues, surrounding cells by these particular pits, and forms the plasmodesmata. And thus they help in communication and the transportation of material from one cell to the another. So cell to cell as well as cell to the space in between, that is actually supported by the parenchyma tissue. Further, how can we compare the nucleus? In the case of parenchyma tissue, nucleus is present, and if the nucleus is present, what property should come in your mind? That it should be a living cell. Meristematic tissue, they are also living cells and they are having a prominent nucleus. Here in this case, the difference is that the nucleus is actually shifted towards the side of the wall or towards the corner of the plant cell. In the case of your meristematic tissue, it was not so. It was centrally located and was very prominent. Here, why it is shifted towards the side? It is because of the presence of vacuole. A vacuole is large, and this large vacuole pushes this nucleus towards a side, and this is why the nucleus appears to be inconspicuous. So further, you can see these nucleus and vacuole here in the diagram. The nucleus is present here in this part. You see, this part, this green highlighted part is actually a nucleus, and this light green shade shown here is actually a vacuole, which is pushing the nucleus towards the side of the cell, towards the boundary of the cell. Besides this, regarding intercellular space, a very important point. In the case of parenchyma tissue, enough space is present. You remember what happens in the meristematic tissue? The meristematic tissue do not possess the intercellular space. They are compactly arranged. But here, intercellular space is prominent. It is present here in between the cells. You can see these are the sheet of cells shown here, spherical cells. You know, if there is a space, then definitely it is going to be filled by either fluid or by the air. So these spaces in between actually supports filling up by the air, so or the different material. So these spaces basically help in the exchange of gases between these area, that is space, and the interior of the cell. If this is not present, then how the diffusion process can take place? How the gaseous exchange can take place? How the different material can be transported? Right? So this intercellular space in between is basically meant for the diffusion of different things like gases, as well as transport of various other material from one cell to the another cell. Now you can see here in the diagram the cytoplasm is shown. So in this case the cytoplasm is of course present, but it is not so dense as it was there in the case of meristematic tissues. Meristematic tissues, we said they are having a dense and abundant cytoplasm. Why? Because they are in the multiplication phase. They keep on forming those particular materials, due to which the cytoplasm appear to be dense. Here it is not that much dense. Here it is different from that. But in this case, the cytoplasm of parenchyma tissue, it contains various cytoplasmic inclusions. It contains secondary metabolite. So this is the difference between the cytoplasm of parenchyma and that of the meristematic tissue. So all these are actually providing different type of benefit to each kind of cell. Further, now you think where such type of tissues must be present. Meristematic tissue are present in the growth region, where the new organs or the new cells are being synthesized or they are generated. So where this parenchyma is present, basically in simple terms, we can say that parenchyma is basically a filling material. It is filling the space between the different kind of tissues present inside the plant. For example, it is present in the cortex of roots. You know what is cortex of roots? If we compare the TS of a leaf, if you just assume the cortex of this thing, then we can say it must appear something like this. If this is a TS, then we can say that they are actually having different regions here. In the center part, there is what? There is a cambium or the vascular bundles, and all those material are located. Besides that, the outer layer is known as what? It is known as epidermis, and this inner layer is known as endodermis. And in the center part, that is the pith, there is the presence of vascular bundles and all other material. So the region between the epidermis and the endodermis, that is known as cortex. So these are located in the particular cortex region, where it is in between this epidermis and the endodermis. We can just write it here also. This outer boundary is what? It is epidermis. Just a second. See, this is epidermis, the outer line, and this inner line is actually endodermis. And the region in between these two layers, there is a distribution of different kind of cells, out of which one of them is parenchyma. And what this parenchyma, this region is known as? It is known as cortex. Now this must be present even in the stems of, section of stem also. If we see the TS of this stem, then of course the ground tissue, this major region of this stem or filling material of this stem is actually made up of parenchyma tissue. Now where else these parenchyma can be located? They are present in the leaves, where in the mesophyll region of the leaves, as well as it is present in some parts of the flowers also. So we can see this diagram here. This will just show you where these parenchyma is located. This is the structure of a leaf. You can see this here, it is a leaf, and this particular region is a section of a leaf. So in this case, let us look at the different regions now. So a leaf is made up of different parts. This outer layer, they are the, we can say, outer covering of the leaf. These are known as epidermis. So the uppermost layer of the leaf is epidermis, and of course at the bottom side also you can see this brown line here. This is known as a lower epidermis. So between exterior or externally to the upper epidermis, it is covered by another protective layer which is known as cuticle. Now in between the upper epidermis and lower epidermis, it is comprised of a tissue which is known as mesophyll tissue. So these mesophyll tissue, they are basically made up of parenchyma tissue. You can see these green structures here. They are chloroplast containing cells. These are forming different structures here now. In the upper side, they are little bit compact, and of course they are having intercellular space in between, but comparatively towards the lower epidermis, the intercellular space is much higher. So due to this particular reason, the parenchyma which is distributed here is actually taking different shape and structure. The one which is making the which is associated with the upper epidermis here is known as palisade parenchyma. These are cylindrical in nature and they have intercellular space in between. Whereas the bottom part, they are more and more they are made up of spherical parenchyma cells, and they are having great intercellular spaces in between. So now this particular parenchyma which is having enough space in between makes it spongy. So such type of parenchyma is known as spongy parenchyma. And together these tissues are known as mesophyll tissue. Which tissue? The one which is present in between the upper epidermis and in the lower epidermis. This mid part of sandwich is known as a mesophyll tissue. Now this is not alone parenchyma. It is having different other kind of tissues also. So those we will study later when we start with the chlorenchyma. Right now, just understand that these parenchyma are present in between the mesophyll tissue also. Now, continuously we are saying that they are having the spaces in between. And where we are studying these parenchyma or the space, it is in the leaf. Now just can you think that how this space is basically supporting the functions of the plants? It is because, see, you know, leaf is the major site of photosynthesis. They are having chloroplast containing cells, and therefore they are able to photosynthesize. That means they are able to synthesize the glucose molecules. Now for photosynthesis, what do we need? Since very early classes you are studying that photosynthesis means the production of food material from carbon dioxide and water in chlorophyll containing cell by utilizing the solar energy. Right? So as we are saying that fine, chlorophyll is present within the cell. Solar energy is provided by the sun, then remaining carbon dioxide and water. Water will be transported by the xylem tissue from the soil. Now what is remaining here is, that is carbon dioxide. Carbon dioxide is present in the environment, in the atmosphere. So these carbon dioxide are supposed to move inside the cells. How? So what happens is, the air from the stomata will fill this particular intercellular space in the mesophyll tissue, and from there the carbon dioxide will be transported into the interior of the cells. And of course then what will happen? These parenchyma which are containing the chlorophyll will absorb the carbon dioxide and photosynthesize the glucose molecule. During this reaction, oxygen is produced which will be diffused out in the intercellular space and then towards the outside. So this is how these parenchyma are playing a major role inside the cell, although they are not supporting the growth, but they are supporting the photosynthesis in this particular leaf. In the cortex of the root, it was helping in maintaining the structure of the root. It is helping in the storage of different things. So this is how it is so important for the function of the parenchyma. Now we can quote it here, that the major function is that they are responsible for the storage of food material. Not only the food material, also different kinds of material. For example, the excretory material, the material which has to be thrown out of the cell, which is not useful for the plants. So that is to be eliminated. So temporarily they are going to be stored in the parenchyma tissue, and then they will be excreted out by various methods. Besides that, it is also helping in the storage of food material which is synthesized by the photosynthetic reaction. So what is synthesized by the photosynthetic reaction? Glucose. But do you really think that glucose is a storage form of food? No. This glucose will be transformed into the starch, and then it is going to be stored in the parenchyma tissue. Further, this stored food in the leaves is not going to reside forever in the leaves only. They have to be transported to different parts of the plant. So from the leaves, they will be transported to the other particular site. Further, these cells, they also help in the healing of various wounds and origin of the adventitious structures. So, you know what is adventitious structure? These adventitious means generation of new organs from the site of not of its origin. Basically what happens, if there are the root cells, they form the roots. If there are the leaf cells, they form the leaves. But suppose you must have seen that roots might arise from different tissues also, from different type of organ also. Like sometimes these roots are grown over the stem region or the trunk region. So that is actually supported by the parenchyma tissue. Now these can also help in the photosynthesis process only when it is containing the chloroplast. Why we are saying this thing? Because it is not necessary that all the parenchyma tissue or the parenchymatous cells are containing the chloroplast. The one which is containing the chlorophyll or the chloroplast, of course they are going to support the photosynthetic reaction. So further, these cells, they are actually metabolically active. So, and secondly, they are having intercellular spaces in between. So this property actually provides the advantage of diffusion processes. It allows the gaseous exchange. It supports not only the photosynthetic gaseous exchange, but also it supports the respiratory exchange of the gases. Besides this, these are actually providing support to the plants. They are providing support to the stem, as they are the major section or the major part of the ground tissue. So they provide support. Also, they help in the transport of material from where? Cell to cell or cell wall and the cell of the parenchyma cell. In short, we can say they are helping in the communication process between the different parts of the plants. Now, depending upon special qualities, the special features, they may be of different kinds. As we are saying that they are having chloroplast, they can be supporting the photosynthesis, then they may be helping in different other forms of function also, as we can see in the form of their types. So the specialized type of parenchyma, they are number 1, chlorenchyma. Which cell is known as chlorenchyma? It is of course parenchyma, but it contains what? Chloroplast. Now you can imagine if chloroplast is present, it can support the process of photosynthesis, and this we have studied earlier. So you can see the structure here. These are palisade mesophyll, these are spongy mesophyll. There are intercellular spaces which are supporting the gaseous exchange during the process of photosynthesis. Now further, if they are forming the palisade parenchyma, Now these palisade parenchyma, they are the type of chlorenchyma only, but they are now elongated. They are the mature cells. These elongated cells provide support to the leaf structure, as well as it helps in the process of photosynthesis. You remember the location in this diagram? We said that they are located below the upper epidermis. So these elongated structure not only help in the photosynthesis, they also help in the storage as well as providing support to the leaf structure. Next is aerenchyma tissue. So what are these aerenchyma tissue? You know, from word aren, can you imagine something now? Aren means air. So these parenchyma cells, they are basically having the lots of air cavity in between. That means what? In short, we can say the intercellular space is very high in case of aerenchyma tissue. Now how this aerenchyma tissue can have lot of air now, intercellular space, how it can increase? It is because these parenchyma cells are having the radiating arms. It means they are star shaped more or less. Now if they are star shaped, then how it is going to be appear here? Something like this. It may be something like this. These are radiating. So due to this particular arm formation, the intercellular space must increase. Another cell, let us say it is located somewhere here. So the intercellular space between these boundaries should of course increase. So due to which this intercellular space is very high. And therefore we are saying that these are known as aerenchyma. Now just imagine where these must be present and what could be the significance. These are basically found in the petiole of banana. You know what is petiole of banana? Petiole is basically a stalk of a leaf. It is a site or a base from where the leaves are actually arising. Suppose this is a plant and from here this leaf structure is actually arising. So this base, this is actually known as petiole. So they are located in the basic petiole of the banana. You know how this banana tree appears? So that must be very clear for you. Then next, they are present in the aquatic plants, and here they are abundantly present. Now what advantage can they provide to these aquatic plants? They provide buoyancy. You know what is a buoyancy? It is actually property which supports the floating of a substance on the surface of the water. So if the air space is high, the object becomes light, and as they become light, they can float over the surface, and this floating property is known as buoyancy. So these aquatic plants, they are able to float on the surface of the water because of the presence of aerenchyma tissue. These aerenchyma tissue are thus abundantly present in the aquatic. This is all about your parenchyma tissue. Next we are going to begin with the chlorenchyma. So till then, thank you so much. We'll meet you soon in the next session. Have a nice day. Good day.